Hi, hello viewers. Welcome back again to one more of the lecture session from this channel. So in this lecture session, we will be studying regarding the procedure of DNA fingerprinting in detail. So here the technique of DNA fingerprinting for the very first time, it was explained by Alex Jeffries. Alex Jeffries for the very first time, he had explained the technique of DNA fingerprinting in detail. And here Alex Jeffries, he had taken the advantage of a short stretch of the DNA sequence which repeats many times, and here it is termed as what? Repetitive sequence. And here the specific repetitive sequence which was considered by Alex Jeffries, here it is nothing but what? VNTR. So here VNTR stands for variable number of tandem repeats. So here why it is termed as variable number of tandem repeats? Because as the name itself suggests, the number of the specific repetitive sequence, it remains vary from chromosome to chromosome in an individual. So the reason why here it is termed as variable number of tandem repeats. Is it clear? So let me write it on the board. The technique of DNA fingerprinting was initially explained by Alex Jeffries. And here Alex Jeffries, while performing the DNA fingerprinting, he had taken the advantage of a short stretch of the DNA sequence which repeats many times. And here the specific short stretch of the DNA sequence which repeats many times, here it is termed as what? Repetitive sequence. And here the specific repetitive sequence which was considered by Alex Jeffries, here it was nothing but what? VNTR. So here VNTR stands for variable number of tandem repeats. So here why it was termed as variable number of tandem repeats here? Because the number of the specific repetitive sequence, it remains vary from chromosome to chromosome in an individual. So the reason why here it is termed as variable number of tandem repeats. Is it clear? So let me write it on the board. Alex Jeffries had taken the advantage of short stretch of the DNA sequence. Termed as repetitive sequence. And the concerned repetitive sequence which was considered by Alex Jeffries, here it was nothing but what? Variable number of tandem repeats. So let me write it on the board. Variable number of tandem repeats. And it is simply, here it is abbreviated as VNTR. So here the specific VNTR, it is a part of mini satellite. Is it clear? The specific VNTR, it belongs to a specific class termed as mini satellite. As I have already explained you, here we can differentiate the specific satellite DNA into two different types. And these may include microsatellite and the other one is mini satellite. So here the specific VNTR, which is a type of repetitive sequence, it belongs to the mini satellite. Is it clear? VNTR belongs to the class of mini satellite. And here the size of the specific VNTR, it ranges around 0.1 to 20 kilobases. Is it clear? The size and as well as the number of the VNTR, it remains vary from chromosome to chromosome in an individual. And here if we consider the size of the VNTR, the size of the VNTR, it ranges around 0.1 to 20 kilobases. Size of VNTR ranges around 0.1 to 20 kilobases. And in the same way, if we consider the number of the specific VNTR, it also remains vary from chromosome to chromosome in an individual. In case of one particular chromosome, as we all know, in case of humans, all the cells, they are diploid. And here one particular diploid cell, it may contain 23 pair of chromosomes or 46 chromosomes. So here, in case of all the 46 chromosomes, the specific VNTR repetitive sequence is observed. And here the number of the VNTR repetitive sequence, it remains vary from chromosome to chromosome in an individual. Number of VNTR repetitive sequence remains vary from chromosome to chromosome in an individual. Chromosome to chromosome in an individual. And here we need to understand that in general, in case of all the individuals, here the specific number of the VNTR repetitive sequence, it remains vary. The number of the VNTR repetitive sequence, it remains vary in case of all the chromosomes in case of one particular individual. But here, the number of the VNTR repetitive sequence, it won't remain vary in case of monozygotic identical twins. In case of monozygotic identical twins, both in case of the monozygotic identical twins, the number of the VNTR repetitive sequence which is observed, it remains very similar. So the reason why both the monozygotic identical twins, they remain very similar in the means of phenotypic appearance. In case of monozygotic identical twins, here the number of the VNTR repetitive sequence, it remains very similar. In case of all the chromosomes, in case of the monozygotic identical twins, the number of the VNTR repetitive sequence, it remains very similar in case of all the chromosomes in case of the monozygotic identical twins. But whereas in case of the rest of the other individuals, the number of the VNTR repetitive sequence, it remains vary from chromosome to chromosome in an individual. The number of VNTR repetitive sequence does not remain vary in case of monozygotic identical twins. And here, because of the rapid advancement in genetic engineering or rDNA technology, the specific DNA sequence which has isolated from any particular gene in case of one particular individual, it can be manipulated as much as we require by taking the advantage of a specific technique termed as polymerase chain reaction. So here, by taking the advantage of polymerase chain reaction, here we can manipulate the number of the particular gene. So here the specific gene which has isolated from one particular living organism, its number can be manipulated by taking the advantage of a specific technique termed as polymerase chain reaction. Is it clear? Polymerase chain reaction, which is simply abbreviated as PCR. Most commonly used to manipulate the number of consent gene which has isolated. Is it clear? And right now we will be studying regarding the various stages which are involved in regulating the DNA fingerprinting in detail. So let me arrange the things here. Look at the specific steps which are involved in regulating the procedure of DNA fingerprinting. So here the very first step here it is nothing but what? Isolation of genomic DNA. So here the specific DNA which has isolated from any specific source in case of one particular individual, it can be used for regulating the procedure of DNA fingerprinting. And the next specific step here it is nothing but what? Digestion of genomic DNA by the means of restriction enzymes into multiple fragments. So let me write it here. Isolation of genomic DNA. And the next specific step here it is nothing but digestion of genomic DNA into multiple fragments by restriction enzymes. And the next specific step here is nothing but what? Transferring of DNA fragments into the electrophoretic chamber. Transfer of DNA fragments into the electrophoretic chamber. And the next specific step here is nothing but what? Separation of DNA fragments by taking the advantage of agarose-gel electrophoresis. Separation of DNA fragments by agarose-gel electrophoresis. And here the next specific step here it is nothing but what? Transferring of DNA fragments from the electrophoretic chamber onto the surface of nitrocellulose membrane or nylon membrane. And here the specific step here it is termed as what? Blotting mechanism. And here the specific blotting which is carried out here it is termed as southern blotting. Is it clear? Transfer of DNA fragments from the electrophoretic chamber onto the nitrocellulose membrane or nylon membrane. And here the specific process here it is termed as what? Southern blotting. And here the last step which is involved in regulating the procedure of DNA fingerprinting here it is nothing but what? Visualization of the specific VNTR probe by taking the advantage of VNTR radio-labeled probes. Is it clear? On the surface of the nitrocellulose membrane, here we can visualize many number of VNTR probes by taking the advantage of VNTR radio-labeled probes. So here these VNTR radio-labeled probes exactly or accurately they bind to the VNTR probes which are present on the surface of the nitrocellulose membrane. So here once when the VNTR radio-labeled probes, once when they accurately bind to the VNTR probes which are present on the surface of the nitrocellulose membrane, these VNTR radio-labeled probes, they emit radiation. And here the specific procedure, here it is termed as what? Autoradiography. Is it clear? So let me write it here. Visualization of DNA fragments, visualization of VNTR probe. So here the meaning of probe is nothing but what? Short oligonucleotide sequence. So here the specific repetitive sequence, here it is termed as probe. So here visualization of VNTR probe by taking the advantage of, by the means of VNTR radio-labeled probe. VNTR radio-labeled probe. So here the particular VNTR radio-labeled probe, accurately it binds to the VNTR probe which is present on the surface of the nitrocellulose membrane. So here once when the VNTR radio-labeled probe, once when it binds to the VNTR probe which is present on the surface of the nitrocellulose membrane, it emits radiation. And here the specific process, here it is termed as what? Autoradiography. So let me write it here. Autoradiography. Autoradiography. Is it clear? So here all these are the various steps which are involved in regulating the procedure of DNA fingerprinting. So here the procedure of DNA fingerprinting, it has many wider applications. So here prior to the explanation of the applications of the DNA fingerprinting in detail, at first let us have a look on one of the specific example which has mentioned in your NCERT textbook. So here by taking the advantage of the concerned example which has mentioned in your NCERT textbook, easily we can understand the procedure of DNA fingerprinting. Is it clear? So let me arrange the things here. And here, in order to understand the procedure of DNA fingerprinting, here we can take the advantage of one of the common example which has mentioned in your NCERT textbook. So in one particular crime scene, C is the concerned victim. So here in order to identify whether C is the concerned son of or the concerned daughter of either the parent A or parent B, here they have carried out the DNA fingerprinting procedure. So initially they have considered the 3 chromosomes, and these may include chromosome 7, chromosome 2 and chromosome 16. In case of all the 3 chromosomes, they have considered both the paternal chromosomes and as well as the maternal chromosomes, because as we all know, in case of humans, all the cells they are diploid in nature. That means the chromosomal content is 2n. One chromosome arrives from the father and the other chromosome arrives from the mother. So the reason why here they have considered both the paternal and as well as the maternal chromosomes. And here by taking the advantage of DNA fingerprinting procedure, here we can conclude that C is the concerned son of or C is the concerned daughter of parent B but not with parent A. So here you make it a small clarification. How far you are able to say that C is the concerned son of or C is the concerned daughter of parent B but not with parent A. If you look at here, the repetitive sequences which were present in case of all the 3 chromosomes, in case of individual C, they were matched with the repetitive sequences in case of all the 3 chromosomes of individual B but not with individual A. Is it clear? The repetitive sequences which were identified in case of all the 3 chromosomes, in case of individual C, they were matched with the repetitive sequences which were identified in case of all the 3 chromosomes, in case of individual B but not with individual A. So in this way, by taking the advantage of DNA fingerprinting procedure, here we can conclude that C is the concerned son of or C is the concerned daughter of parent B but not with parent A. And if you look at here, on the surface of the nitrocellulose membrane, the specific repetitive sequences which were identified in case of individual C, they were matched with the repetitive sequences of individual B but not with individual A. So in this way, by taking the advantage of DNA fingerprinting procedure, here we can conclude that C is the concerned son of or C is the concerned daughter of parent B but not with parent A. And as I have already mentioned you, DNA fingerprinting, it has many wider applications. So here what are the various wider applications of DNA fingerprinting here? DNA fingerprinting, it is most commonly used in case of forensic sciences. Is it clear? So let me write it on the board. Applications of DNA fingerprinting. Applications of DNA fingerprinting. It is most commonly used in case of forensic sciences. Forensic sciences. That means in order to identify the criminals, in order to identify the rapists, and even in order to identify the parental disputes, here DNA fingerprinting, it plays an important role. Then in the same way, in order to regulate the genetic diversity and as well as speciation, DNA fingerprinting, it plays an important role. So let me write it here. Genetic diversity and speciation. That means in order to differentiate the two people who belong to two different continents, here DNA fingerprinting, it plays an important role. So here by taking the advantage of DNA fingerprinting, here we can differentiate the two people who belong to two different continents. And here in order to understand how the DNA fingerprinting procedure, it has solved many of the complicated cases in our country, here we can take the advantage of one of the common example which was took place earlier. So here once when Rajiv Gandhi assassination was took place in the year 1991, so here in order to identify some of the body parts of Rajiv Gandhi in the concerned location, some blood samples were collected from Rahul Gandhi and as well as Priyanka Gandhi. So here within the blood samples, the DNA sequence which is present, and within the DNA sequence, the repetitive sequences which were identified in case of Rahul Gandhi and as well as Priyanka Gandhi, they were matched with some of the body parts which were identified in the concerned location. So in this way, by taking the advantage of DNA fingerprinting, the body parts of Rajiv Gandhi, they were identified in the concerned location during his assassination. Is it clear? So here this is regarding DNA fingerprinting from the molecular basis of inheritance in detail. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.